Yeah, so I was doing my PhD in human immunotherapy and did a lot of monitoring of the immune system in cancer patients. And a professor at Stanford at that time who really, I think we all believe he's going to get the Nobel Prize for the discovery of the receptors on T cells, Mark Davis recruited me, and I was also doing part of my med school years, and so I dropped out med school, have finished my PhD, and moved to Stanford to really try to reinvent the way we study immunology. And so for probably 150 years, we've been using animal models, and for the most part, these don't translate into human biology. So we decided to focus on human studies, to do more of that type of research. And so when we were doing the 1,000 Immunomes project that we started in 2007, really, so a while ago, that I'm going to allude to in a moment, the one thing that we found repeatedly over and over were aging signals. We're looking at immune system, and then we're seeing aging signals, and that turned my entire research to aging. And I thought we should use the human immune system to understand aging. And there's a very clear connection here.
In one of your interviews that I listened to, you said to your father one day that you wanted to have a massive impact on human health, to have a big impact in the world. And you were thinking about med school, and you went to med school for a few years. But your dad gave you some really sage advice. Right. What was that advice?
Yeah, well, the advice was really that when you treat patients, you do it in a piecemeal fashion, one by one. And you can create impact, but not to the extent that you're interested in what you're communicating to me now. It seems like you want to have more impact in the humanity, as opposed to thousands of people. In the best case scenario, as being a doctor, those real doctors that do medical work, right? But in my case, with a PhD, then I thought, okay, this could expand the avenues and really have a massive impact on things that all medical doctors could use to help more people and have more impact.
Absolutely. I think your dad was very wise to send you in that direction, if you like. And okay, now let's focus in a little bit on the 1,000 Immunomes project and immunology. And I've been fascinated in the last couple of years with, in my studies of the hallmarks of aging, I kept coming across, inflammation, immune system dysregulation, thymic involution, all of these things that happen as we age. And of course, my audience know of my battle with my mum to help her with all her comorbidities that she has, and struggling with her immune system, and especially going through cancer treatments and so on and so forth. And so it really came apparent to me, and when I discovered the 1,000 Immunomes project and started reading your research, I'm like, yeah, this is it, because the immune system is the key foundation of slowing the aging process. The paper that you put out, I think it was in 2023, about chronic inflammation is the cause or at the base of all the hallmarks of aging. And that really rung true with me as being like, these are affecting all things. So let's start with the immune system first. How did you were you seeing age-related markers when you were studying immunology? How did you come to that?
Right. Really, this idea that the immune system and certain aspects of the immune reaction, like inflammation, can drive aging processes is rather new, right? In terms of the history of immunology, we've known that the immune system and the inflammatory response will help you clear viruses and infections, other sorts, and if it goes awry, you develop autoimmunity. We've known that for many, many decades, but only in the early 2000s, we started looking at some aspects of immunology that were somewhat accelerating biological aging in different systems, right? And so then the idea of studying the immune system to look for determinants of aging processes and mechanisms and hopefully develop different interventions to target the immune system and extend health span and a lifespan by default. It was kind of the focuses of the of the 1,000 Immunomes that
you sort of alluded to something before, which was that, you know, all the immune system science to date has been mostly in animal studies, and the animal studies just really don't translate, and that we've got this sort of a problem. It takes 10 plus years to develop a drug, $2 billion or something to bring a drug to market. 90% of our drugs fail. We're just not getting, you know, where we need to be with that model. And so we decided to focus then onto to actually studying human immune systems. That's quite a shift, you know, because usually we start with mice or C. elegans or something like that, and then move to mice and then move to humans. Why is your approach, like, to go straight to working on humans, really quite revolutionary?
Yeah. You know, this is really something I didn't start. It was really a trend in the field to better model the human biology. We have been in this revolution of technologies that now we have to extensively monitor every single aspect of human biology, from, you know, all the genes that are in the genome, all the proteins that are in the proteome, all the metabolites. We're talking about hundreds of thousands of molecules from blood. So it is really a good time for us to start thinking about, you know, monetizing, if you will, on those technologies to, or capitalize to really understand the immune system in humans. As you alluded to, you know, there's almost no good model for human disease, let alone human aging. I mean, it's really, really poor models. All the animal models that we see, from C. elegans to all the way to animals like mice, or even, you know, non-human primates is probably the closest that you can get. But we're using humans, and we're using human organoids that are mini, brains or mini hearts or mini immune systems. We develop in the lab to really have the genetic and the phenotypic variation that we see in human beings, and not be, looking at the wrong place. Although, you know, we've used animals to really dig into the mechanism, because oftentimes with human studies, it is challenging to understand mechanistic behaviors. But for the most part, those even those do not translate as well. So I decided to really focus on human biology, biopsies, blood, in large studies, and measure everything under the sun, and then try and use AI, machine learning, to cope with the data and get significant insights.
Yeah,
into
because
the
this is
biology
a
and health
convergence of
disease.
technologies, haven't we? We've got computational, sciences, and we've got all these exponential technologies all happening basically at the same time in history, which gives us the opportunity now, like never before, to be able to do these things. Like you couldn't do the computations that would have been required previously, because you just wouldn't have had the manpower to do that. And now, you know, computers, they can do things in seconds what would have taken days. So this has accelerated the rate that we can research. Or decades.
Oh, well,
yeah.
it would have taken decades.
Wow.
Yeah, well, it would have taken decades. Honestly, the power to store is not minor and analyze and compute now is basically unprecedented. Anything that you want, you can do today. We're using these supermassive language models and to, extract relevant information from 30 million papers in half an hour in the lunchtime.
You'd be like a kid in a candy store sometimes when you think about, I mean, I know even with my, basic knowledge of very basic things, and I can do things that I couldn't even do 3 or 4 years ago, just with ChatGPT, let alone the stuff that you guys have available to you, it's just really going to revolutionize. I mean, I hope, that within 5 to 10 years time, we're going to be sitting in a completely different world with completely different paradigm of the way things are done. My fear is that, the clinics aren't keeping up. Like, we're already not. We're already,